quick review that reactions come in two flavors. Well, many, but two main flavors. Energy storing and energy releasing, right? And either way, there's going to be a hill to climb. Either way, there is a distance between the reactant's energy level and the energy level we're going to have to get to make the reaction go. That's called the activation energy. And then the distance between the reactants and product's energy is either the energy stored or released. And that enzymes, their deal is, energetically their deal is, they reduce that activation energy. So with an enzyme, your activation energy is going to be lower. Fine, that's how we graph it. But what's happening in the real world, we said, is that enzymes actually have this little specially shaped nook called the active site, this place in this big protein, this folded up chain of amino acids, where the substrate, which remember is what we rename the reactants when an enzyme's involved, that's where the substrates fit like a lock into a key. I drew you one version of it, and then we had a little animation of the other version. The basic idea is it fits in there, and then the enzyme's active site puts just the right kind of pressure on either the two things that need to smush together or the one thing that needs to split apart so that that can happen more easily. So that's what it physically looks like when the energy stuff is going on that you drew. Do we have any questions about that so far? Anything you kind of looked at last night and you're just like, I don't understand what that means? So then we mentioned that temperature can be a factor for enzyme activity. And we talked about the one case where that can be good, just when you have a fever, your white blood cells work better. But mostly temperature, we don't change much on purpose. pH, we keep very steady. That the way we typically regulate our enzymes, and that most living things do, is using other enzymes or other protein fragments. And the one example I want to show you of that, and this is going to be kind of a, I'm not going to write it out for you. I'm going to show you a sort of a narrated video clip. I'm going to have you try to put it into words yourself for this last concept. That's what I'd like to do. So it's called competitive inhibition. The proteins, sort of mini enzymes that you make to deactivate some enzymes are called competitive inhibitors. Remember that enzymes are catalysts, meaning they increase the rates of reaction without getting used up. And so if you were able to watch a reaction happening with an enzyme present, you'd see the reactants, and you'd see the enzyme come in, and again, you'd see the enzyme grab the reactants, smush them together, create the products. Grab another set of reactants, smush them together, create the products. And a bunch of enzymes would be present, each of them doing that over and over. The simplest way I can teach you that your body makes something that slows down a reaction, that deactivates an enzyme, is the short story is you basically make a blocker molecule. It's called a competitive inhibitor, and it is enough of the same shape as the reactants that it'll stick in the active site, but it doesn't do anything. It doesn't react to form anything. In fact, all it really does is keep real reactants from plugging into that active site. It's a blocker molecule. So let me show you that in this video.
During the normal enzyme catalytic cycle, the substrate encounters an enzyme with a specific active site to which it binds, forming an enzyme-substrate complex.
This is normal.
The enzyme then facilitates the breakdown of the substrate to its products, which part from the enzyme, leaving the active site free to catalyze another substrate as the cycle begins again.
Can't. That's as loud as it goes here.
Competitive inhibition occurs when an enzyme encounters a blocker, which mimics the properties of the substrate and binds to the enzyme's active site. Thus,
that plugged in, substrate can't plug.
The active site is not available for attachment, and no reaction will occur.
So that's competitive inhibition. There's also non-competitive inhibition,
Non-competitive,
which you don't have to know, but for what it's worth, it's where the protein fragment that you make doesn't plug into the active site, it plugs into a different spot, but that's enough to change the shape of the active site so the active site doesn't work. Don't worry about it. You'll get there in AP biology. If you take that, you'll talk about all those different types of inhibition. and regulation of enzymes. Yeah?
Is there ever more than one active site on an enzyme?
You know, you could sort of consider that second spot there almost like a second active site. I don't know specific examples where they're both called active sites, but it very well could be. That's a good question. There could be more than one active site, as far as I know. Other questions? Okay, so that really is the last chunk of new material, except tomorrow I'm going to bring in some stuff, and we're going to play with a real enzyme a little bit. It won't be the kind of lab you're used to because of our rules about touching the same stuff, but it'll be more of a demo lab where you'll see some enzyme activity, and then we'll learn some stuff about temperature and enzyme activity. It won't be a lot, okay? Just kind of a fun thing to do. But that really is about it. Your test is the day after tomorrow. Your sample essays are due tomorrow. Okay. So, all right, let's see.